So the next chapter we are going to discuss, that is respiration in plants or cellular respiration, and the weightage is normally around 2 to 4 questions in our Neet examination. And in cellular respiration there is aerobic and also anaerobic respiration. Aerobic respiration is the complete oxidation of respiratory substrate or glucose in the presence of oxygen and produces 6 molecules of carbon dioxide, 6 molecules of water and normally 36 molecules of ATP. 36 molecules of ATP energy releases. And aerobic respiration completes in 3 different stages. Aerobic respiration, the process of aerobic respiration completes in 3 different stages. The first stage is called as glycolysis. Glyco, which means glucose or carbohydrate, and lysis is the splitting. So glycolysis is a splitting of glucose into 2 molecules of 3 carbon compound called pyruvic acid. So 1 molecule of glucose or C6H12O6 break down into 2 molecules of 3 carbon compound called as pyruvic acid. So the process of glycolysis that completes in the site of glycolysis is cytosol or cytoplasm, and glycolysis is common to both aerobic and anaerobic respiration. The only stage that is common to both aerobic and anaerobic respiration is glycolysis. Glycolysis is also called as EMP pathway because the process was discovered by Emden, Meyerhoff and J. Parnas. So that's why it is called as Emden-Meyerhoff-Parnas pathway, or it is called as EMP pathway. So the another name of glycolysis is EMP pathway because it was discovered by Emden, Meyerhoff and Parnas. Emden, Otto, Meyerhoff and J. Parnas. And the site of glycolysis is cytosol or cytoplasm, and it is common to both aerobic and anaerobic respiration. And here 1 molecule of glucose undergo breakdown through series of enzymatic process and that end in the production of 2 molecules of pyruvic acid. 1 molecule of glucose, a 6 carbon compound, break down into 2 molecules of 3 carbon compound pyruvic acid. And the second stage after glycolysis in the aerobic respiration, the pyruvic acid will enter into the next process that is called as Krebs cycle, and Krebs cycle is also called as citric acid cycle because the first stable compound produced in the citric acid cycle or Krebs cycle is citric acid, and it is also called as tricarboxylic acid cycle because it has 3 carboxylic group, and that's why it is also called as TCA, tricarboxylic acid cycle. So Krebs cycle is also called as citric acid cycle because the first stable compound is citric acid, and it is a tricarboxylic acid, that's why it is called as tricarboxylic acid cycle or TCA cycle, or are same. And the site of Krebs cycle is mitochondrial matrix, the process that is taking place only in the mitochondrial matrix, and it is present only in the aerobic organism, and as a result of Krebs cycle there produces NADH plus NADH++H+ and FADH2 are mainly produced as a part of Krebs cycle. And the third stage, that is the terminal oxidation on electron transport chain. Terminal oxidation on electron transport chain, and here the second reactant or of the aerobic respiration, oxygen, is utilized, and it is taken place in the inner membrane of mitochondria, and as a result the NADH or FADH2 will be converted into ATP molecule, and it is the terminal oxidation, and the ATP synthesis is taken place in the electron transport as the part of electron transport chain or terminal oxidation. So this is the 3 different stages of aerobic respiration, and in this chapter cellular respiration or respiration in higher plants we are discussing the 3 different stages: along with this, we will discuss about anaerobic respiration and some aspects of respiratory ratio. Then the chapter will be finished. So we can move into the glycolysis in detail. Glycolysis is the first step of aerobic respiration, and glycolysis is also called as EMP pathway, that is Embden-Meyerhof-Parnas pathway. The schematic representation of glycolysis was discovered by Embden, Meyerhof and Parnas. That's why it is called as Embden-Meyerhof-Parnas pathway. And the site of glycolysis is cytoplasm or cytosol. And here one molecule of glucose, a 6-carbon compound, through the series of enzymatic process break down into 2 molecules of 3-carbon compound pyruvic acid. And glycolysis is common to both aerobic and anaerobic respiration. Glycolysis starts from glucose C6H12O6. It is 1 molecule of glucose and it is a first 6-carbon compound. And here in the initial stage, the first stage, the enzyme involved in glycolysis is the first step of glycolysis is hexokinase. And here, this is the skeleton of a glucose molecule. 1, 2, 3, 4, 6 carbon. And this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon and 6th carbon. 1 molecule of ATP that is present in the cytoplasm break down into ADP and inorganic phosphate, and this phosphate will be joined with the 6th carbon and forms glucose 6-phosphate. And in the initial stage of glycolysis, it is mediated by the enzyme hexokinase, and hexokinase is the enzyme that starts the aerobic and also anaerobic respiration. This is the first enzyme, and here 1 molecule of ATP break down into ADP plus inorganic phosphate. That inorganic phosphate combines with the 6th carbon and forms the glucose is converted into glucose 6-phosphate, and it is a 1 molecule. And in the next step, that is glucose 6-phosphate converted into fructose 6-phosphate, and it is only isomerization. Glucose and fructose are isomers. So the next stage, glucose 6-phosphate is converted into fructose 6-phosphate. And the next stage, here 1 more molecules of ATP break down into ADP and inorganic phosphate. Now this ATP is break down into ADP plus inorganic phosphate, and this inorganic phosphate combine with the first carbon. So normally in the initial stage, glucose is converted into glucose 6-phosphate with the help of enzyme hexokinase and 1 molecule of ATP, and next stage glucose 6-phosphate undergo isomerization and forms fructose 6-phosphate, and in the next stage the fructose 6-phosphate converted into fructose 1,6-diphosphate, and this is also 1 molecule, this is also 1 molecule. And these are the initial stages, and these are the charging of the preparation for the glycolysis or the charging for the glycolysis. That is here 1 molecule of glucose, and the second 1 molecule of glucose undergo phosphorylation with the help of enzyme hexokinase and forms glucose 6-phosphate, and the glucose 6-phosphate undergo isomerization and form fructose 6-phosphate, and fructose 6-phosphate again undergo 1 molecule phosphorylation by 1 molecule of ATP and converted into fructose 1,6-diphosphate. And this is the charging, and this is the preparation for the glycolysis. And the next stage, the fructose 6-phosphate undergo breakdown. We know that fructose 6-phosphate is a 6 carbon compound containing molecule with 2 phosphate at 2 ends, and it will be break down and produces 2 isomers with the same number of carbon, same number of hydrogen and same number of oxygen and phosphate. And these 2 molecules are, one is dihydroxyacetone phosphate. It is a 3-carbon compound and that is DHAP, dihydroxyacetone phosphate. And the second molecule, fructose 1,6-diphosphate, undergo cleavage or breakdown and produces one molecule of dihydroxyacetone phosphate, a 3-carbon compound, and another is glyceraldehyde 3-phosphate, and it is also a 3-carbon compound. It can be G3P. It is a short form. And these two molecules are with a 3-carbon compound and they are triose phosphates. And these are isomers with the same number of carbon, hydrogen, oxygen and phosphate, and it can be interconvertible with the help of enzyme. And as per NCERT, in the part of glycolysis, we have to study only one enzyme, that is hexokinase. Each steps are mediated by the enzyme, but we are not mentioning here because as per NCERT, we have to study only one enzyme, that is called as hexokinase, the initial enzyme. So glucose is converted into glucose 6-phosphate with the utilization of one molecule of ATP. Then glucose 6-phosphate is converted into fructose 6-phosphate through isomerization, and fructose 6-phosphate again phosphorylate and form fructose 1,6-diphosphate, and the cleavage, this is the cleavage of fructose 1,6-phosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Next is the process will be continued through the subsequent sequences or the stages of the glycolysis was continued through glyceraldehyde 3-phosphate, but this dihydroxyacetone phosphate will be converted into glyceraldehyde 3-phosphate and continue the process. So the coming each and every stages are we must be considered twice, because one for glyceraldehyde 3-phosphate and another for dihydroxyacetone phosphate. So here we have glyceraldehyde 3-phosphate. It is a triose phosphate, 3-carbon compound, and it is interconverted with dihydroxyacetone phosphate through the cleavage, and glyceraldehyde 3-phosphate will be continue the process. So the initial stage, the first, next stage, glyceraldehyde 3-phosphate undergo a partial oxidation, that is the removal of hydrogen, and that will be produced 1,3-bisphosphoglyceric acid. It is 2 molecules of 1,3-bisphosphoglyceric acid is produced, and along with this process it is a oxidation process. So one molecule of NAD plus is converted into NADH plus H plus. The energy value of one molecule of NADH plus H plus is equivalent to 3 ATP, that we will study in the coming stages. And it is a 3-carbon compound, 1,3-bisphosphoglyceric acid, and the 1,3-bisphosphoglyceric acid in the next step, that is here it is with first carbon and also third carbon I have phosphate, that is 1,3-bisphosphoglyceric acid, and 1,3-bisphosphoglyceric acid that releases the phosphate from the first carbon, and this phosphate combined with ADP and form ATP molecule. So in the next step, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglyceric acid. 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceric acid, and that is 3-PGA, and the same biomolecule we are discussing in as a part of C3 cycle, the first stable compound produced in the Calvin cycle. So here 1,3-bisphosphoglyceric acid that releases the phosphate from the first carbon, and that inorganic phosphate combined with ADP and form ATP molecule. ADP combined with inorganic phosphate and forms ATP molecule. This is the first ATP molecule produced as a part of glycolysis. This is the first ATP molecule produced as a part of respiratory process. So 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceric acid. It is also 2 molecule. And next step, 3-phosphoglyceric acid is converted into 2 phosphoglyceric acid. It is also a isomerization. Two molecule. Third carbon has phosphate, and it will be shifted into second carbon, that is 2-phosphoglyceric acid. And the 2-phosphoglyceric acid that releases one molecule of water, and it is converted into phosphoenol pyruvic acid, that is PEP, and it is also a 3-carbon compound. 2 molecules of phosphoenol pyruvic acid is produced. And in the next stage, phosphoenol pyruvic acid that has a phosphate and that releases phosphate, and that phosphate combined with inorganic phosphate and produces ATP, and then the phosphoenol pyruvic acid is converted into pyruvic acid, and it is also a 2 molecule. So here it is starting from glucose. 1 molecule of glucose is involved in the glycolysis, and the end product is 2 molecule of pyruvic acid, and this is the initial stage of cellular respiration in all organisms, both aerobic and anaerobic organism, and this is the base of life in organism, because when any organism that take organic compound or respiratory substrate that have to release the energy through the different stages like aerobic or anaerobic process, and the aerobic respiration that involve glycolysis, Krebs cycle and electron transport chain. As a part of glycolysis, there are 10 different stages. First is glucose undergo phosphorylation with the help of enzyme hexokinase and converted into glucose 6-phosphate. Then fructose 6-phosphate isomerization, fructose 1,6-diphosphate again phosphorylation, and cleavage of fructose 1,6-diphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And the glyceraldehyde 3-phosphate continue the process, and the glyceraldehyde 3-phosphate in the initial stage there is an oxidation process, partial oxidation, and the releases proton, and it is converted into 1,3-bisphosphoglyceric acid. 1,3-bisphosphoglyceric acid. First carbon and the third carbon has phosphate, and the first carbon containing phosphate is released, and that combined with ADP and form first ATP molecule. This is the first ATP produced as a process, first ATP produced as a part of glycolytic process or any respiratory process, because glycolysis is the initial stage. 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceric acid. 3-phosphoglyceric acid converted into 2-phosphoglyceric acid, a position change, positional isomerism, and then 2-phosphoglyceric acid releases 1 molecule of water, and that results in phosphoenol pyruvic acid, and then phosphoenol pyruvic acid releases the phosphate, and that combined with the inorganic phosphate and forms pyruvic acid. So this is the different stages, and here there is an interconversion between dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So glucose is converted into glucose 6-phosphate. Glucose 6-phosphate is converted into fructose 6-phosphate. Fructose 6-phosphate is converting to fructose 1,6-diphosphate. Then cleavage, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. They are interconvertible, and dihydroxyacetone phosphate always convert into glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate continue the process. Glyceraldehyde 3-phosphate convert into 1,3-bisphosphoglyceric acid, and 1,3-bisphosphoglyceric acid into 3-phosphoglyceric acid, 2-phosphoglyceric acid, phosphoenol pyruvic acid, and then pyruvic acid. So here we can see the first is the charging of the glucose, that is the glucose is enriched with phosphate for the initiation of the process, and this is the first. These stages are first one, second one and third one. These stages are the part of charging, and then cleavage and deriving ATP. In the coming stages, NADH, ATP and ATP are produced in the different stages. So this is the brief of a glycolytic process that is taken place in both aerobic and anaerobic organism. The site of glycolysis is cytosol or cytoplasm, and glycolysis is also called as EMP pathway, Embden-Meyerhof-Parnas pathway. And next is what is the fate of pyruvic acid in different organisms, because in each organism the fate of pyruvic acid is different, and we are studying the aerobic respiration. As a part of that, there will be anaerobic respiration also. So what will be happen the pyruvic acid in different organisms, and that is the next one. So as a result of glycolysis, that produced 2 molecules of pyruvic acid. And what is the fate of pyruvic acid? We know glycolysis is common to both aerobic and anaerobic respiration, and the pyruvic acid will be changed in 3 different ways in different organisms. In yeast-like organism, that is in Saccharomyces yeast, the pyruvic acid converted into acetaldehyde, a 2-carbon compound and 1 molecule of carbon dioxide by the presence of enzyme pyruvate decarboxylase. Pyruvic acid converted into acetaldehyde, a 2-carbon compound plus 1 molecule of carbon. Pyruvic acid is a 3-carbon compound and acetaldehyde is a 2-carbon compound and 1 carbon is released as carbon dioxide. And it is mediated by the enzyme pyruvate decarboxylase. And the acetaldehyde then converted into ethanol by the utilization of 1 molecule of NADH plus H plus is produced as a result as a part of glycolysis, and this acetaldehyde is converted into ethanol, and it was mediated by the enzyme alcohol dehydrogenase enzyme. And this process is called as alcoholic fermentation. And the net result of ATP in the alcoholic fermentation is 2 ATP. 2 ATP is the net production of ATP during the alcoholic fermentation. And in the case of lactobacillus, the bacteria, and also human muscle cell on exercise, the pyruvic acid will be converted into lactic acid. Pyruvic acid is a 3-carbon compound and also lactic acid is also a 3-carbon compound, and it was mediated by the enzyme lactate dehydrogenase. And here also 1 molecule of NADH plus H plus is converted into NAD plus molecule, and that is the reducing power is utilized. And here also the net gain of ATP is 2 ATP molecule. 2 ATP molecule, and this is called as lactic acid fermentation. In both cases there is no utilization of oxygen, and that's why it is called as anaerobic respiration. And here the pyruvic acid either converted into ethanol, as in the case of yeast-like organism, with the help of 2 enzyme pyruvate decarboxylase and alcohol dehydrogenase enzyme, or pyruvic acid can be converted into lactic acid, as in the human muscle cell during the laborious exercise, that is in the insufficient of oxygen, and also lactobacillus, it is converted into lactic acid. But in the case of all eukaryotic cell with mitochondria, the pyruvic acid will be enter into mitochondria and undergo a series a process that is called as oxidative decarboxylation, and the 3-carbon pyruvic acid is converted into a 2-carbon compound that is called as acetyl coenzyme A. Acetyl coenzyme A enter into a series of chemical reaction that is called as Krebs cycle, and produces, as a result it produces carbon dioxide and water, and it is also related to ETS or electron transport chain, and produces 36 molecule of ATP, the net gain 36 molecule of ATP, and that is taken place inside the mitochondria. This is aerobic respiration. So the fate of pyruvic acid that is produced as a result of glycolysis is different in different organisms. That it may be converted into acetaldehyde and ethanol, as in the case of alcoholic fermentation, or pyruvic acid may be converted into lactic acid in the lactic acid fermentation, or in the eukaryotic cell as a part of aerobic respiration it will be enter into mitochondria and undergo the first step is oxidative decarboxylation and Krebs cycle, and Krebs cycle is clubbed with electron transport chain or terminal oxidation and produces 36 molecule of ATP. So next we are moving to the aerobic part, that is decarboxylation and Krebs cycle. So in the eukaryotic organism, the pyruvic acid enter into mitochondrial matrix and it undergo oxidative decarboxylation. Oxidative decarboxylation is also called as link reaction, because it link between the anaerobic phase, that is glycolysis, and the aerobic phase, that is Krebs cycle. So oxidative decarboxylation is also called as link reaction. In oxidative decarboxylation, the pyruvic acid that enter into mitochondrial matrix, and in presence of NAD plus and magnesium 2 plus ion, and coenzyme A and pyruvate dehydrogenase enzyme, it will be converted into a 2-carbon compound that is called as coenzyme A plus, and they releases one molecule of carbon dioxide and NADH+. And here the acetyl coenzyme A that is produced as a result of oxidative decarboxylation that combine with oxaloacetic acid. Oxaloacetic acid is a 4-carbon compound with acetyl coenzyme A. It is a 2-carbon compound, and acetyl coenzyme A is a 2-carbon. Oxaloacetic acid is a 4-carbon compound, and acetyl coenzyme is a 2-carbon compound. And with one molecule of water, it is a condensation reaction, and it is mediated by the enzyme citrate synthase and produces citric acid. Citric acid. It is a 6-carbon compound, and that's why the Krebs cycle, or it is also called as citric acid cycle, and citric acid is a tricarboxylic acid. That's why it is called as tricarboxylic acid cycle or TCA cycle. And next is the stepwise removal of carbon dioxide, and it is associated with the stepwise removal of proton and coupled with the formation of NADH+, H+, and then the next stage, the one molecule of carbon is released and it is converted into citric acid, releases one molecule of carbon because it is a 5-carbon compound, and it will be converted into a 5-carbon compound. It is called as alpha ketoglutaric acid. So citric acid is a 6-carbon compound, and citric acid is formed by the presence of the enzyme mediated by the enzyme citrate synthase, and citric acid undergo decarboxylation, and along with decarboxylation one NAD+ molecule is converted into NADH+H+, and it is a 5-carbon compound, alpha ketoglutaric acid. Again it releases one molecule of carbon dioxide. There also one NAD+ molecule is converted into NADH+H+, and here the alpha ketoglutaric acid is a 5-carbon compound. When one carbon dioxide is released, it will be converted into a 4-carbon compound, and that is called as succinyl coenzyme A, because here one coenzyme A is added. And succinyl coenzyme A releases the coenzyme, and it is coupled with one GDP molecule combined with inorganic phosphate and converted into GTP, and succinyl coenzyme A will be converted into succinic acid. And succinic acid is converted into, succinic acid is a 4-carbon compound, and it is converted into another 4-carbon compound that is called as fumaric acid. And during this step, one molecule of FAD+ is converted into FADH2, and here acetyl coenzyme A, a 2-carbon compound combined with oxaloacetic acid, a 4-carbon compound, along with the water, undergo condensation, and it is mediated by citrate synthase, and it results in the formation of citric acid, a 6-carbon compound, and it releases one molecule of carbon dioxide along with the synthesis of one NADH+H+, and produces a 5-carbon compound called alpha ketoglutaric acid. And next is again one molecule of carbon dioxide is released, and it is converted into succinyl coenzyme A by addition of coenzyme A, and succinyl coenzyme A is converted into succinic acid by the release of coenzyme A along with a substrate level phosphorylation that is called as the GTP formation, and succinic acid is converted into fumaric acid. With during this stage, one FADH2 is FAD+ is converted into FADH2, and fumaric acid is converted into malic acid, and malic acid is converted into oxaloacetic acid. During this stage, one NAD+ is converted into NADH+H+. This is the schematic representation of Krebs cycle. or citric acid cycle or TCA cycle. And here citric acid 6 carbon compound, alpha ketoglutarate acid is a 5 carbon compound, succinyl coenzyme A succinic acid 4 carbon compound, fumaric acid, malic acid, and again oxaloacetic acid. So here any of the stage there is no formation of ATP directly. Only one stage GTP is produced as a part of substrate level phosphorylation, but all other stages NADH molecules are produced, the N and FADH2 molecules are produced. The energy value of 1 NADH plus H plus is equal to 3 ATP when they undertake the aerobic stage, or it is called as electron transport chain or terminal oxidation. It results in the formation of 3 ATP, and FADH2 results in the formation of 2 ATP. So NADH is equivalent to 3 ATP and FADH2 is equivalent to 2 ATP. So as a result of the one molecule of glucose that undergo the complete aerobic respiration, glycolysis, Krebs cycle, and electron transport chain, and that produces total number of 38 molecule of ATP, that is a gross production. But we know the initial stage 2 molecules of ATP are utilized, so the net result is net ATP production, that is 36 molecule of ATP. And this ATP are produced only when it is passed through, the molecules are passed through, the protons and electrons are passed through electron transport chain. So as a part of Krebs cycle, acetyl coenzyme A combined with oxaloacetic acid produces citric acid into alpha ketoglutarate acid into succinyl coenzyme A into succinic acid, fumaric acid, malic acid, and again oxaloacetic acid. The number of carbon atoms and which step the NADH is produced, how many stages, 1, 2 and 3 and 4 stages inside the mitochondria that produces NADH plus H plus from one molecule of acetyl coenzyme A, but actually there is 2 molecule of acetyl coenzyme A or 2 molecule of pyruvic acid from 1 molecule of glucose. So when we calculate the total ATP, the gross production is 38 molecule of ATP and net gain is 36. This is the outline of aerobic and anaerobic respiration. And study the NCERT textbook very detailed, and do the maximum questions related to the chapter, and be prepared for the coming our Neet examination.
Bright Education Center, Medical and Engineering Entrance Coaching Center, Hilal area, Doha, Qatar. For further enquiries, contact 306000679.